Good morning, everyone. I'm Joe Thrush. I'm a fourth year PhD student at the Rosalind Institute in London, near Big Rock, and also at King's College London in the UK. And this is part of my PhD titled Investigating the Membrane Fusion Events of Nipah virus. So what is Nipah virus? Nipah virus is a zoonotic hematovirus in the Paramyxophilidae family. It was discovered in 1999 in a patient presenting with encephalitis. The patient typically presents with fever, dizziness, headaches, and this often develops into severe encephalitis. The mortality rate is extremely high, between 40 to 100% across the outbreaks, and outbreaks vary between 1 and 260 cases, mostly occurring in the Indian subcontinent and the Southeast Asian countries. Transmission is from bats, particularly fruit bats, to humans, and this is either directly to humans or via pigs. On the map here, we have countries orange with hematovirus cases, with blue stars indicating Nipah virus cases, mainly in India and the Southeast Asia regions. However, the red dots indicate positive serum samples of hematovirus DNA in bat urine. Nipah virus is an 18 kilobase negative 7% single-strand RNA genome, and it has 6 structural proteins, of which the main two to store are the F protein, which is the fusion protein, and the G protein is the glycoprotein. The glycoprotein binds the host cell receptor F and B2 and coordinates fusion with the F protein. And it's a process that's not really understood in the literature so far. So there are two models that describe this interaction. The first is the clamp model, and this postulates the G protein stabilizes the pre-fusion F, i.e. interactions are occurring before receptor binding. And the second model is the provocateur model, and this postulates the G protein activates the pre-fusion F, i.e. interactions occurring after receptor binding. And there's a whole host of evidence in the literature for either one of these models, as well as potentially a temporal evidence in prolonged receptor binding. So my work has been focused on unpick which of these models is true. Because Nipah virus is obviously incredibly dangerous, it's categorized in BSL-4. We use two models in the lab to describe it. The first is the pseudovirus model, and this uses the two Nipah virus proteins, the F and the G, as well as the HRE1 core of GAG and POL, and a reporter plasmid of luciferase. So you add pseudovirus to cells, you wait 48 hours, and you get a readout of luciferase expression. The second model is this VLP model, where we have the two F and G, as we mentioned, and the M or matrix protein. And here we have a beta-lactamase enzyme fused to the M protein, and by addition of the VLPs to cells, we can measure cleavage of a fluorophore, and that determines how much fusion of the VLP is occurring. This talk is split into two parts. We first have cryo-electron tomography of the VLPs, and particularly to interrogate the F and G interactions. And the second part is the generation of anti-Nipah antibody, or single-domain antibody, which comes from camelids. And two reasons for this is to develop a novel therapeutic prophylactic or point-of-diagnostic reagent, because there's no current licensed therapeutics or vaccines available for Nipah virus. And the second is to use the neutralization of these antibodies to interrogate the membrane fusion events of Nipah virus and viral entry. So tomography of VLPs looks like this. They're very pleomorphic. They're either spherical or bulbous, and they vary between 20 to 800 nanometers in size, and they're heavily decorated with both F and G. On the right-hand side here, you can see that the F forms this single layer of protein, while the G forms this double layer. And they appear to kind of separate on the surface of the VLPs. We next performed sub-kilogram averaging of the F protein. So the bottom here we have a double layered membrane, and rather than a single isolated NIV-F trimer, we're starting to notice we have NIV-F dimers of trimers, and they're interacting in a pre-fusion conformation on the surface of the VLPs. In particular, the fusion peptide, which is responsible for insertion into the host membrane, is hidden inside the actual interface of the F proteins. If we increase the threshold of our volume, I want to look at the tilting of the two proteins relative to the membrane. So this is being resolved with C1 symmetry to look at how each of the individual trimers bend relative to the membrane. And from the side, both of them are fairly perpendicular, parallel to each other, and normal to the membrane. However, from the end, one of them, the blue one, is tilted about 7.5 degrees to the right, and the red one is tilted about 17.5 degrees to the right. So they're twisted about 25 degrees relative to each other. And we've attributed this twisting ability to protein 486, which is between the alpha helices of the X domain and the alpha helices of the transmembrane domain. And this allows the X domain to kind of move about on the surface of the VLP and reach out a host membrane. By increasing the threshold further of our volume, we can start to see other structures appearing nearby. In particular, we can see 4 more NIV-F proteins that are arranged in a triangle relative to the central 2. Obviously, this is an averaging technique. So in the bottom right, the central 2, the ones marked A and B, we have about 100% of our subtomograms. On the right, the E and F is about 60, and the C and D have about 10%. So this is not a lattice where you expect 100% occupancy at all sites. This is more like a disjointed assembly network. We can put this all back together. So we have our Nipah virus in the tomogram, where we have our double-layered membrane of F and G. We then can put in the membrane itself that's been segmented along with the positions of the subtomograms of this NIV-F dimer. And by zooming in, we can see that prior to the previous slides, we're not getting a single dimers and trimers, we are getting this extended network across the surface of the VLP and the pseudoviruses. So it appears to be a kind of transient interaction, holding them in a metastable pre-fusion conformation, ready to be released by the G protein compartment of the host receptor. Second part of this talk is looking at developing an anti-NIV-F nanobody. So to do that, we've got the NIV-F in a pre-fusion conformation, and we've immunized a llama, waited 3 months, and isolated the white blood cells, or PBMCs, of which we've generated a VHH library from the mRNA and done 2 rounds of phage display and picked out our strongest binders using a screening ELISA. We then sequenced them on small scale expression, turn them into the FC vector to make them bivalent constructs, and then characterize them with neutralization and fusion inhibition assays from the 2 models I mentioned earlier, and characterize the epitopes using cryo-EM. This is the protein we've used to immunize. It's just the X domain of the NIV-F, stabilized with 4 point mutations in the trimerization domain. And this is a funnel diagram showing the number of those we have each step. So we start with about 200 in our screening ELISA, whittling it back down to 12 that we express recombinantly as FCs, and only 3 that we do cryo-EM work with. Of the 12 that are expressed as FCs, they're split into 2 major epitopes. We have the first epitope of 10, which includes the best fusion inhibitors and neutralizers. We have a smaller epitope just of 2, which is the worst fusion inhibitors. And the rest of the talk, I'm going to focus on just 3 of them. So F10 and D2 are the neutralizing ones, and then G10 is the non-neutralizing. So for D2 and F10 neutralizing, we've characterized their binding to the X domain with BLI. So we have association and dissociation on each of the graphs of an antibody relative to the purified protein. Both D2 and F10 bind with low to subnanomolar affinity as monomers. However, as FCs, they bind much stronger as expected. In terms of their functionality, they inhibit fusion of the VLP with subnanomolar IC50 values, and they neutralize the pseudovirus with subnanomolar IC50 values comparative to a published antibody against the G protein. We then characterize the epitopes using cryo-EM. So there's 2 structures here. The first one on the left is the NIV-F, which has been incubated with the D2 antibody, and the second one is the NIV-F incubated with the F10, the other neutralizing one, and then the G10 non-neutralizing. As expected, the D2 and F10 share the same epitope that's confirmed by BLI studies, whereas G10 is off to the side. We can put this back together with looking at the NIV-F dimer of trimer structure we saw from the subtotal averaging. We have our NIV-F dimer epitope in light green, and we have our nanobody epitopes in blue, and they don't overlap. However, by putting in the positions and superimposing the structures of D2 and F10, we can see that they actually sterically block the formation of this NIV-F dimer. So addition of these to the VLPs to the pseudovirus will break apart this network that we're seeing on the surface of the VLPs. So So in summary, we've done the sub tomogram averaging of the NIVEC on the VLP in a pre-fusion confirmation, and we've seen these NIVEC interactions at high density. We've also seen it shown they form this extended oligomeric network, and we're generating antibodies against NIVEC with high levels of fusion inhibition and neutralization. There's two epitopes present we found, there's only one on the VLP. And the antibodies we've generated sterically block the dimerization of this NIVEC dimer. So I thank you all for listening. We've recently published this in our archive, and if you have any jobs, I'm available in 4 months.